Hello, I am Torsten Wittmann from the Department of Cell and Tissue Biology at the University of California in San Francisco. Today, I would like to tell you about our recent work on using optogenetics to locally and acutely control microtubule dynamics in live cells, to ask questions about how the microtubule cytoskeleton shapes cell morphology, dynamics, and function. I also would like to place this in the context of neuron development and morphogenesis. As vividly illustrated by this image of differentiating stem cells, neurons are some of the morphologically most complex cells in our bodies, and microtubules play a critical role in generating this complexity. For example, this image shows the outline of a very young cortical neuron differentiating from a human-induced pluripotent stem cell. As you can easily see, microtubules, shown here in white, invade every single one of the nascent growth cones branching from this neurite. Many neurodevelopmental disorders are caused by proteins related to the microtubule cytoskeleton, and in fact, there is now even a name for such diseases that are caused by mutations in tubulin itself. Microtubules are cylindrical hollow polymers of alpha-beta tubulin dimers that are arranged head to tail in protofilaments. In most mammalian cells, 13 protofilaments are arranged in a staggered helix to make up the microtubule wall. GTP-loaded tubulin dimers polymerize onto the microtubule plus end, and GTP is subsequently hydrolyzed in the microtubule wall. GTP hydrolysis is associated with a conformational change longitudinally along tubulin protofilaments, moving adjacent tubulin dimers closer together. GTP and GDP-PI-loaded tubulin subunits are thought to protect the growing microtubule plus end from sudden depolymerization. The structure of this protective cap is complicated and still not completely understood, but recent cryo-electron tomography data have challenged older ideas in the field and now strongly suggest that microtubules grow as individual curved protofilaments that subsequently close into a tube. Cells control microtubule polymerization dynamics and microtubule network organization on many levels. For example, CCAP5 or XMAP215 is a microtubule polymerase that facilitates the addition of GTP tubulin dimers to growing protofilament ends, and by itself, CCAP5 accelerates polymerization of pure tubulin by 5 to 10-fold. Other proteins specifically recognize the microtubule wall conformation associated with the tubulin guanosine nucleotide state. End-binding proteins, or EBs, are thought to bind to the GDP-PI tubulin conformation near the growing microtubule end, while the neuronal microtubule-associated protein double cortin recognizes the GDP microtubule lattice. This difference in specificity is shown here in a cell expressing M-cherry-tagged EB1, displaying the characteristic comets at growing microtubule ends in purple, and GFP-tagged double cortin in green that binds along GDP microtubules and does not overlap with EB1 comets. Double cortin, of course, is well known for its function in brain development, as a multitude of double cortin mutations can cause lissencephaly spectrum diseases, in which new neurons do not properly migrate through the developing cortex. However, the molecular function of double cortin in developing neurons is not well understood. A few years ago, we found that double cortin specifically recognizes straight GDP microtubules, and our more recent work in neurons indicates that double cortin is only present on straight microtubules in growth cones. We hypothesize that double cortin-mediated mechanical stiffening of these microtubules may be important for growth cone advance, but this is a different story for another day. Instead, I want to now focus on our efforts to control protein complexes at the end of growing microtubules that depend on plus-tip protein interactions with EB1. EB1 is a dimer with C-terminal calponin homology domains that recognize the growing microtubule end structure. The N-terminal EB homology domain has a hydrophobic pocket that binds disordered protein regions containing SXIP peptide motifs that are also surrounded by positive charges. There are about 40 such so-called plus-tips in mammalian cells with diverse and often poorly characterized functions that are recruited to growing microtubule ends through interactions with EB1. Because in cells, microtubules grow on the order of 20 micrometers per minute, we wanted to develop an optogenetic tool to control plus-tip protein networks at growing microtubule ends with high spatial and temporal accuracy. While there are several plant and microbial photoreceptors that can be harnessed for optogenetic approaches, the LOF2 domain of oat phototropin 1 that in plants mediates blue light phototropism has several advantages. The LOF2 domain is small, only requires a flavin mononucleotide cofactor that is present in all eukaryotic cells and reverts to the dark conformation relatively quickly. Upon absorption of a blue light photon, the major conformational change is an undocking and unfolding of the J-alpha helix at the C-terminus of the LOF domain. We further used an optogenetic system pioneered by the Hahn lab, in which a protein AZ domain was selected by phage display to bind with high affinity to the LOF2 domain dark state, but this LOF2 C-dark complex rapidly dissociates in blue light. This is seen in this proof of principle experiment, in which purified LOF2 and C-dark molecules only form a complex in the dark, but this complex is absent when we run a native gel while exposed to blue light. To generate a photo-inactivated EB1 variant that we named PI-EB1 and that could replace EB1's function as a central plus-tip complex organizer in the dark, we inserted the LOF2 C-dark module between the EB1 N and C-terminal domains with the expectation that blue light would result in dissociation of the plus-tip adapter from the microtubule binding domain, resulting in disassembly of the plus-tip complex at growing microtubule ends. This project was mostly work by a talented postdoc, Jeffrey van Haren, who recently started his own group at the Erasmus Medical Center in the Netherlands. Indeed, in an experiment in which we transiently expressed both halves of PI-EB1 tagged with different fluorescent proteins, blue light had no immediate effect on the N-terminal domain that directly binds to growing microtubule ends. In contrast, the C-terminal half very quickly dissociated from growing microtubule ends with a half-life of less than 200 milliseconds. Importantly, the very rapid blue light-induced PI-EB1 dissociation, together with dark reassociation kinetics on the order of seconds, allows for subcellular control of plus-tip complex dissociation with high spatial and temporal accuracy. This experiment of a cell expressing PI-EB1 with only the C-terminal part tagged with M-cherry illustrates this by alternating blue light exposed regions between the top and the bottom of the cell. M-cherry C-dark EB1 C rapidly, locally and reversibly dissociates from growing microtubule ends only in these blue light exposed regions. With this graph showing the blue light illumination profile and relative EB1 C plus end association when the top half of the cell is exposed to blue light, and this graph showing on and off switching of EB1 C plus end binding in the two alternating regions over time. On the other hand, if we tag the N-terminal half of PI-EB1 with M-cherry, we can use this to analyze the effect of PI-EB1 dissociation on microtubule polymerization dynamics. This movie shows 20-second time-lapse sequences of the same cell before, during, and a few minutes after blue light exposure, demonstrating reversible inhibition of microtubule growth as indicated by a substantial decrease of the microtubule growth rate, the duration of growth events, and the overall number of growing microtubules. Lastly, it is important to note that PI-EB1 is not dominant negative and that in this and subsequent experiments, endogenous EB1 is removed by either RNA interference or CRISPR genome editing, and that in this case, all other plus tips that we have tested also dissociate from microtubule ends. This brings up the question of how cells achieve physiological microtubule growth rates. In vitro, purified tubulin polymerizes much slower than microtubules grow in cells, and as I mentioned earlier, while the microtubule polymerase CCAP5 can accelerate microtubule growth, by itself it is not sufficient to support microtubule growth rates typically seen in mammalian cells. However, because there is published evidence that there are interactions between CCAP5 and the plastid protein network, we next tested if CCAP5 localization to growing microtubule ends depended on EB1. However, this experiment shows that in contrast to EB1-dependent plastids, which dissociate from growing microtubule ends as a result of pi-EB1 photo-dissociation, the dynamics of CCAP5 in cells appear unchanged. This is further supported by our observation that CCAP5 remains on microtubule ends even in cells in which both EB1 and EB3 have been deleted by CRISPR genome editing. In addition, microtubule-associated CCAP5 looks very different and much more punctate compared with typical EB1 comets, and in data not shown here, CCAP5 localization correlates very poorly with EB1 on growing microtubule ends. These data and the current model that microtubules grow as curved protofilaments prompted us to think that two different structural transitions on growing microtubule ends must occur together for optimal microtubule growth in cells. By itself, tubulin polymerizes slowly at rates of 1 to 2 micrometers per minute. While CCAP5 catalyzed protofilament elongation can increase the microtubule growth rate, microtubules can only grow as quickly as protofilaments close into a tube, and the formation of protofilament lateral interactions becomes rate limiting. EB1, on the other hand, binds between protofilaments and may promote protofilament zippering, but does not increase the rate of tubulin dimer addition. Only if both structural changes are optimally coordinated, microtubules grow at the high rates exceeding 20 micrometers per minute observed in mammalian cells. I've just shown you that pi-EB1 photodissociation inhibits microtubule growth, but how does this affect the microtubule network as a whole? In this pi-EB1 cell, microtubules are labeled with M-cherry tubulin. Blue light exposure is globally turned on when the image on the left appears that shows the GFP-tagged C-terminal half of pi-EB1. Comets at the microtubule ends can be briefly seen before pi-EB1 dissociates. Soon after the blue light is turned on, microtubules begin to shorten and disappear from the cell periphery. Note that not all microtubules are affected equally, possibly indicating microtubule populations with different stabilities. While it is difficult to determine the amount of polymerized microtubules in a cell, it is quite easy to measure unpolymerized tubulin fluorescence. And the increase of unpolymerized tubulin in this graph shows that the microtubule network reaches a new equilibrium within a minute. This is even more evident in a cell expressing dominant active RAC1, a small Rho GTPase that induces cell edge actin polymerization and retrograde flow. Microtubules are rapidly cleared from the right side of the cell when it is exposed to blue light, and this induced microtubule network polarity can be maintained for an extended period of time. Although it is known that microtubules polarize in most migrating cell types, such that the microtubule organizing center and the bulk of microtubule growth are oriented toward the direction of migration, it is less well understood if this microtubule network reorientation is a cause or a consequence of directed migration. We therefore used our pi-EB1 cells to address this question. The blue square in this video is not a physical barrier, but simply an image of the reflected blue light pattern. Nevertheless, this pi-EB1 cell turns around every time it enters a blue light zone and remains trapped within this virtual box for more than 8 hours. This and other experiments indicate that EB-dependent plastid networks are required for directional movement, and I invite you to read our paper for more detail. The pi-EB1 experiments that I have shown up to this point were all in a cancer cell line and relied on knockout of the endogenous EB1 gene and plasmid-based expression of pi-EB1 constructs. To adapt this to more complicated cell systems, such as developing neurons, we came up with a genome editing strategy to insert a bicistronic genetic cassette containing the LOV2C-dark module directly into the EB1 gene and thus in one step convert EB1 into a light-sensitive variant. Genomic PCR and immunoblotting simply confirm successful insertion of this pi element, and these images show induced pluripotent stem cell colonies generated in this manner, in the dark and during blue light exposure, demonstrating the expected pi EB1 dissociation at growing microtubule ends. The final experiment I want to show today is work in progress, coming back to our initial question how plastid protein complexes control neuron morphogenesis. This video shows the extending neurite of a newborn cortical neuron differentiating from pi EB1-induced pluripotent stem cells. The neurite growth cone advances along a micropatterned laminin track toward a blue light box. I will let you make your own conclusions here, but would also like to caution that this is currently one of few experiments, and we are currently working to combine such optogenetic assays with chemotactic gradients to test how growth cone guidance is controlled by microtubule placent protein interactions. All that remains now is to thank our funders, the people who are doing the work, and you for listening. I'm happy to receive feedback or answer questions either by email or on our Twitter account. Thank you.